আচ্ছা আপনি তো বেরিয়ে গেছেন নাকি? ও আজকে তো আচ্ছা এক মিনিট কথা বলা যায়? আচ্ছা একদম আচ্ছা কালকে আপনার সাথে বসে ওই লিস্টটা করব। আর এই কি বলে অয়ন পাল 8C বাড়ি বিক্রি করে দিয়েছে না? আচ্ছা ওনার এগেইনস্টে দেখছি ৭২,০০০ টাকা পড়ে আছে।
Hi everyone. We'll wait a couple more minutes for a few more to add, and this would be the last session for the course, at least for me. And yeah. All right. Hi. So we'll start. This is the last session, as I said, and this is a doubt clearing session for the course for the NPTEL course, basic course in an ornithology. And I'm Jobin Varughese, and this is a part of my Prime Minister Research Fellowship, and that's about the introduction. Yes, Govind.
I'm really sorry to say this, sir, but I'm just currently walking home. Could you mind waiting for just 1 minute?
Sure. Yeah. So, did you guys did you guys go through the lecture, the last lecture? I mean, yes, Dave. Yes, Dave.
Okay. Good evening.
Hi.
So I think this is only about the week 12 questions. Right?
Yeah. This is not a revision session.
Yeah. With week 12, I have a basic problem. See, there are some questions to which which were not covered in the lecture.
Okay.
See, I will give you the examples. Question number 20. See, question number.
What was the question number 20? I wouldn't know.
Okay. Question number. See, these are all related to some bird counts, etc., which was supposed to be there in some video, which we never got.
Okay.
See, there are only 2 videos in lecture in week 12, which are on molecular biology.
Yeah.
Molecular techniques.
Okay.
Okay. Now, but these two, like, see, question number 13 was how many photos of rosy starling have been uploaded on eBird Macaulay Library from Gujarat in 2022.
Okay.
Now it says please refer to the lecture and the tools provided.
Okay.
Down to nearest 10.
Okay.
Now I could not find the lecture in which this was covered.
This
I don't know if others have found it.
this
If we
was
can,
probably Ashwin Viswanathan's.
and which is not reflecting there in week 12 or in any other week. At least I can.
No, I think Ashwin's lecture is somewhere there. I have seen it.
I don't know if other participants can also help. Then can.
Oh, it
we
was
can-
more of a yeah. That lecture was more of a, because I remember it happening. It was more live. Yeah, it was a live session.
So, but suppose somebody misses that live session, is a recording available?
Recording might be available. I mean, ideally it should have been up by now, but you still have time for the 12th assignment. No, you don't.
Sir?
No, 12th assignment is already over.
Okay.
So all this discussion is academic or more for learning purposes.
Yeah.
The date was over on 18th.
Yeah, no. So this would be up at least before your exam, definitely, the final exam. But I'm not sure why it was not uploaded before that.
I don't know. See, I mean, so this is my worst performance in this assignments, and basically there are 3 or 4 questions which I could not answer because we just didn't have the source.
But you could have, okay. I mean,
If somebody else has a reference in this group, if they can just post it here, I'll.
Yes, Govind, go on.
You can find the live lecture in YouTube.
Can you post the link in this chat, please?
I just got home just a second.
Yeah, he'll do that mid of the session or something.
Yeah. Okay.
Yeah. All right, I'll start this session with the first question. Yeah. It's on your screen. For all those who have joined recently, this is not revision session of any kind. So yeah, I mean, if this is only restricted to week 12 lectures. There are over 11 people, and I've got like 6 answers. Okay, if you remember lecture 12, the first lecture, Robin showed, if you Google apomorphy or just apomorphy, the first Wikipedia link would show you this image where it clearly shows what apomorphy, plesiomorphy, homology, everything is. So apomorphy is basically, okay, I'll tell you the answer first. So the answer is A. It is unique to a particular clade and all its descendants. So basically, if a trait is present in a particular species and its descendant, but it wasn't present in the ancestors, and the trait is only present in the particular species or individual and the descendant. So that would mean that it is apomorphy, and B is it is an ancestral characteristics and not derived. It is actually a derived or a novel characteristics, and it's not an ancestral characteristics. It would have been a plesiomorphy, would be an ancestral characteristics. It cannot be used to characterize the entire clade. In fact, apomorphy, whatever the trait is, can be used to define the entire clade with so all the individuals or all the species in that particular clade or taxa would have that particular trait. Anyway, we'll this would be further explained in the further questions. So yeah. C is trunks in elephants and D is legs in lizards. Sorry about the typo. It's plural, elephants and lizards. So based on the definition that we talked about in the previous question, apomorphy is a unique characteristics that is present in what is present to a, that is unique to a particular clade and all its descendants. So feathers in birds, all birds have feathers, true, I suppose. Leglessness in snakes, all snakes do not have legs, that's a trait. So trunks in elephants, there are like several species of elephants, like the Asian, African itself has like two species, so or two subspecies, but all the elephants have trunks. And legs in lizards, all the species of lizards have legs?
No, sir.
Yeah,
there are some lizards in which do not have legs. Like I forgot the name. Smooth lizard, something like that.
Yeah, I forgot the name also. But we know the fact.
Glass skins, I think.
Yeah, I think. Yeah. So at least we know by for a fact that there are lizards without legs. So that is not an example of apomorphy. That is not a characteristic that is present throughout the clade. So yeah, that could be a plesiomorphy if it was a characteristics of one of the ancestors as well. So yeah, hopefully this example explains what apomorphy is.
Sir, slow worms. They're called slow worms.
Oh yeah. Thank you.
I think slow worms are snakes. So in all likelihood, that's not the one.
No, actually, slow worms are lizards because they have eyelids. Snakes don't have eyelids, but lizards do. That's how you can tell the difference.
Okay, so I guess the common names are always interchangeable.
I think
Legless
about
lizard may
skinks.
refer to, it's a common name for the family Pygopodidae. They are distinguished from snakes, possessing eyelids, possessing external ear openings, lack of broad belly scales, notched rather than forked tongue, having two more or less equal lungs, and having a very long tail. Okay. While snakes have a long body and a short tail. Okay. Makes sense. So there are several families. Yeah, slow worm is one of the family Anguini. Then there are there are no generic names for all these families, but yeah. All right.
Sir, I have a question.
Ah.
In regards to feathers and birds, you described apomorphy as something that the ancestors do not possess, but all the descendants possess.
Yeah.
In terms of feathers in birds, the ancestors of birds did possess feathers,
You're
right,
saying
sir?
about the dinosaurs having feathers?
Yeah.
Yeah.
So in a way, that too isn't come isn't part of apomorphy, right, sir?
Yeah, it could be
In
said.
a way.
Yeah, in a way. Yeah. Yeah. All right. Third question. Okay, so apomorphy has like okay, apomorphy has like two kinds. Uh, so this kind of apomorphy is found only in one taxon and not found in any other or outgroup taxa, not even the closely related ones. So most of you say C. Let's see what's the answer. It is C. It's odd apomorphy. Only one of the taxon, only one taxon has that particular trait. So let's go to the other question. Yeah.
Sir,
Yeah.
isn't homoplasy and convergent evolution rather similar?
Yeah.
So the having both options is kind of redundant, right, sir?
And you could differentiate. That's a good thing. Anyways, the answer is synapomorphy, which is a trait found in terminal groups of a clade and inherited from a common ancestor for which it was odd apomorphy. So yeah, I mean I can't explain more. All right, so DNA hybridization is one of the earliest ways by which birds were classified as species without referring to the morphological attributes only. So, Sibley and Alquist, 1960, I guess, and yeah, let me give you the answer first. So, it's basically temperature. So what happens is there are DNAs of two species. One which is a probe, which is basically with which another species is to be compared. So then so it's heated. DNAs get denatured, and then after denatured DNA, then annealing happens, and it might happen that one DNA combines with the other DNA. So, at what temperature does this annealed DNA get denatured again? And that is what the hybridization. So. So that temperature depends upon the how similar these two DNAs are. So if they're very similar, the temperature would get higher or something. So
Sorry, sir. What was your last? What did you mention? The last? The more similar the DNA, the greater the temperature.
yeah,
The greater the temperature at which the annealing would happen.
at which the yeah, no, not annealing. The denaturing, it's heated again to denature it again, annealed DNA, and then the greater the temperature, I mean the temperature at which the denaturing happens again is what the temperature used in the DNA hybridization.
To prove how closely they are annealed.
Yeah. Oh. Hey, sorry about that. You guys were not discussing. Wow. Can you hear me?
Yes, yes.
Yes, you are.
Yeah. Sorry about
Yes,
that.
sir.
We have had like these frequent power cuts since the summer started. Yeah. So yeah, the sixth question. Could everyone try the sixth question? Or did you not get the chance to see the sixth question at all? DC. Okay. Okay, this is the sequence of methods from DNA to phylogeny. So what is the exact method? I'll give you a minute more. Do people have molecular biology background over here? Because I don't. Just asking. Yeah, so the answer is D. Most of you are right. Yeah, I think most of you. Yeah. Amarnath says not me. Yeah, me too. All right. So basically, DNA to phylogeny. Of course, the first step is what? Not extraction. The even before extraction, there's a step involved. What is that? The most important step, which is the most difficult step.
Collection.
Yes, DNA collection. So how does that happen? You go to the field, you catch a bird or any animal, but basically this is a bird course. I mean, this is a course on ornithology. So catch a bird through mistnet. That's what we've been doing, and then collect blood samples. Why does the blood sample work in birds, but it might not work in human beings? Any answer? Any, like, it's a trivia, but any reason? It might not work in mammals. Like we have a squirrel lab as well, which is in IISER Tirupati. They don't collect blood samples. They collect tissue samples. Why is that? Like from the tail, tissue from the tail or something.
Maybe RBC don't have the nucleus.
Yes, exactly. So the bird blood has nucleus. It's a nucleated blood, and the mammal blood does not have a nucleus in it. Or the RBCs don't have nucleus in it. That's the limit to what I know about this. So I'll move on to the next question. Govind, the humans complain, birds don't. Please, birds complain a lot. You should hear the kind of scream they make when they're caught. I mean, it's not verbal complaint, but it's more like scream. All right, so whole genome sequencing is basically getting the sequence for the entire genome of the animal, but it's not done. They're done like piecewise, and they're like arranged or they're looked at, and I mean, it's all happening within the vial or within the machine. So depth is basically, I mean, this is to the extent that I can explain. Is depth is basically the answer. Sorry, the answer is reliability. I don't know how sequences came. Answer is B. Okay, so answer is B, reliability. Please don't. Yeah. Basically, so there are these pieces of sequences that are arranged, and then if you the depth would actually tell you ki how many sequences contain the same base pair in where it is supposed to be. So if the number of sequences are high, that means there's a high depth to that whole genome sequencing, and if the number of sequences are low, then there's a low depth to the whole genome sequencing, and the depth would basically tell you what is the reliability of that of the whole genome sequencing, that whole process. So lower depth in WGS would mean less reliable data, higher depth in WGS would mean more reliable data, and it's basically, yeah, it also, I mean, it depends on the sample that you've collected. If the sample is bad, in the sense that it does not have enough DNA to be sequenced, then you'll get a low depth WGS. So, yeah.
Sir, can DNA be found, enough DNA be found from a feather alone?
Yes. So usually, I'm not exactly sure, but people have collected tail feathers. So they pluck a tail feather, and that is the end of that feather is used to. So it's basically a tissue as well. So whatever the feather and the tissue surrounding, obviously the feather itself is a dead cell. So might not give you the DNA, but the tissue surrounding that end of it would actually give you some DNA, and I suppose that is reliable because at times, particularly in roadkills and all, you usually don't collect blood because the heart is not pumping, so the blood can't be collected. So it's usually the tail feather or something of that sort. So all right, so items not added in a PCR. So PCR is polymerase chain reaction. Basically, one DNA strand is undergoes certain changes, and the PCR, what it does is, with one DNA strand, it can actually create a lot more duplicates. So to create duplicates, to create duplicates of DNA, what all do you need? Like DNA is formed of what is the DNA formed of?
Nucleotides.
Yes. So to create copies of DNA, you require nucleotides. So these are the free nucleotides that are added, and primers basically go and exactly anneal with that particular site that is supposed to be duplicated. And then what polymerase does, it's an enzyme that what it does is it pulls in all the nucleotide and starts annealing along that DNA chain, and then it's again heated, and the annealed chain is then separated from the original DNA. Then it becomes two DNA strands. The same process continues, and then it becomes another DNA. I mean, so the same process continues in the second cycle, and so then it becomes 4, and similarly the other cycles continue like that. So if n is the number of cycles, then what is the number of DNA strands formed from one single DNA strand? What is the number of duplicates formed from one single DNA strand, if n is the number of cycles?
2:n.
Yes. Thank you, Astagimat. I think at least, I suppose. Did you know that, or did you know it after the lecture?
No, I knew it. I come from medical background.
Sorry, can't argue over there. All right. So the answer is D, lysis buffer. Lysis buffer has no role in PCR machine. It's usually, lysis buffer is used when the samples are collected, like blood samples. You have to put it somewhere. You can't have minus 80 degrees Celsius fridge or minus 40 degrees Celsius fridge to store the lysis.
It is used in extraction, I think.
Lysis buffer, no, not used in extraction. It's basically used to store the blood sample for Queen's lysis buffer, at least.
Okay.
I'm not sure if there are other lysis buffer. I mean, yeah. So
what we use, one lysis buffer that dissolves all the membranes and separates the.
that's yeah. So there are two processes. There's one more lysis buffer. Yeah. You're right. So the Queen's lysis buffer that we use is used for storing the blood sample on the field. So like it can be stored at room temperature without the blood going bad or the DNA, going denatured or whatever. So, yeah. All right. So the answer, I think people have answered mostly. The answer is, you can do a lot of stuff with genetic data, particularly measure genetic diversity, genetic connectivity, even sex of particular bird species. But yes, Govind, I think you have a doubt.
Well, I know it's not possible to directly measure functional diversity, but it can sort of be used to measure it, right? So,
yes, can you explain that? I'm also curious.
I'm just guessing, but with enough information in an environment, we can match it to particular species and get an idea of the functional traits.
Like how? So you're saying, say one of the functional traits is that the bird is a frugivorous bird. So you're saying you match that functional trait with a particular I don't know. Set of genes, and then you is that like that? And see if
we can match. We can get an idea of the species or family, and we can ascertain whether it would be a frugivore or not, and it's sort of an indirect and not a very foolproof way, but it's still somewhat possible, I guess.
Yes, possible. Yeah.
I think in this case, function depends on the expression of that particular gene.
Yeah.
There will be a gene, but many times it may not be expressed.
Yeah.
So that may be the why the functional diversity cannot be measured actually.
Also, I mean, this is like, I don't know, very random, but have you guys heard of something
Yeah.
called pleiotropy? Pleiotropy. I forgot how it's pronounced, but I suppose this is how it's pronounced, pleiotropy or pleiotrophy. But it's like the same set of genes can be responsible for two very different kinds of functional diversity. I mean, functional traits, or two very different kinds of characteristic traits of that species. So, I mean, that might also like confuse a lot of these things. So,
Even the expression in antibody generation also, there are a lot of what we call as combinations, gene recombinations. So
huh.
exactly the function, what comes out may be different.
Yeah, yeah, yeah. So, anyways, yeah. I mean, it could be true, it could not be true, but yeah, nice discussion. All right. Well, of course, you can do sexing for all these species, but like Govind uses this word a lot, it would be redundant. So, why? I mean, which species would it not be redundant?
The one with the least
Govind.
dimorphism.
Yeah. Why are people? Okay. Yeah, Dave. It's non-dimorphic birds are usually called monomorphic birds. So, I mean, no, I mean you don't have to be sorry about it. It's a term that I mean, of course, it's a term, so it's useful to know these terms. That's all. Sure. So people got the idea. Rose ring parakeet is a dimorphic bird. Grey jungle fowl is like dimorphic to a great extent. Even white drum shama is a dimorphic bird. And I added adults because sometimes the sub-adults don't exactly look very different when it comes to sexes. So that's why I added the adult part. White-bellied cholakili, on the other hand, is a very monomorphic bird, and it's very difficult to tell apart the male and female in the species on field. So even though even though you can tell apart by telling that which individual is singing and which not, even though there have been instances of female singing songs, but still, just to confirm, we I mean our lab also does sexing of blood samples from yeah, sexing of this particular species. All right. So these are Hemosporidia responsible for avian malaria, and Plasmodium, Hemoproteus, and Leukocytozoon. But they are carried, or their vectors are different. So the answer is A. So Plasmodium is transferred by mosquitoes, I think Culex. Hemoproteus is transferred by biting midges, and leukocytosome is transferred by black flies. So, yeah. Okay, so genotypic fitness is basically, can someone define fitness?
The passing on of the success of an organism in survival and passing on its genes throughout different generations.
Yeah. So fitness is basically the success of this individual to pass on its genes to the next generation. So, so genotypic fitness increases with increase in dash loci. Um, okay, the last two are useless. The first two, A and B, are something that we should be concerned about. Um, Between A and B, why do you think heterozygous? I mean, okay, I'll just tell you the answer. So, why do you think heterozygous loci would be much better and homozygous loci would be less, uh, like attribute less to the genotypic fitness? Any answer? Any simple answer? Heterozygous is basically,
Diversity of alleles.
yeah. Heterozygous is basically zygosity of alleles. Yeah. Govin.
The heterozygous alleles would mean two different variations of it.
Yeah.
This would lead to greater diversity and variation, right, sir?
Yeah. Vedant has said larger gene pool confers better adaptability. Yes, so it basically gives a better chance for the individual or the species to survive, or to be able to survive an environment that is possibly changing or something. So although it might not show the benefit at a particular at a particular at a very mild environment, but if the environment changes in the future, then these heterozygous genes might or loci might be able to help better in the survivability of the species, or for that matter genotypic fitness. So heterozygous would mean the alleles. There are several versions of or variations of alleles, and homozygous would mean that there are only only one variation of that particular allele, and that's why inbreeding is deleterious, because inbreeding brings in more homozygosity in the species, so they'll have lesser chances of survival in the future.
Sir, what's the downside of heterozygosity? There must be some kind of trade-off.
There's something called outbreeding. Is this mine? No, it's not mine, right? The voice, huh? There's something called outbreeding. So people know what inbreeding is, where individuals of very similar genetic structure breed together and produce offsprings. There's something called outbreeding, where individuals, so people, there has been example. I think I've mentioned that in one of the questions, where there are this particular mountain goat which was thought to be of the same species, but they were living in different kinds of different habitats, or different basically very different latitude, and then they were brought together because their numbers were decreasing, and so they wanted to increase the heterozygosity of the species, so they were brought together and they were made to breed with each other. But what happened was, there was one population that had produced offsprings at a particular time, so just before at a particular time, so that before the winters the offspring was large enough or adult enough to escape the winter harshness. And then there was another which never had that kind of extreme winter in the locality that it lived in. So when they were bred together, what happened was, it was bred in one of those extreme harsh winters. So all the offsprings were being born at in that particular harsh winter. So most of them died. So yeah, so heterozygosity in that sense, but that was something that the humans intervened in. But otherwise, I don't think there's a problem with heterozygosity as such. Can you think of any other thing?
So what you're saying is, sir, that the heterozygosity might, if it might lead to also lead to like unfavorable traits popping up.
No, I'm not saying that. I'm just talking about the outbreeding part. But heterozygosity it may lead to deleterious. But that's a normal. It's just okay. So if deleterious mutations would happen if it's even if it's homozygous or heterozygous. But what's the problem is, I mean, if you look at it at a population level and not at an individual level, the problem with homozygosity is some of the deleterious basically harmful alleles kind of tend to accumulate within the population, and then they cannot be weeded out. But it can be very easily weeded out in a heterozygous population. So that's one difference.
This is an especially large problem for small isolated populations, right? So like islands, maybe.
Yeah, so that would happen. Yeah, they will happen to be more homozygous populations, these smaller populations.
But I've also read somewhere that small populations like this are somewhat more resistant to the effects of inbreeding. Like they have some kind of resistance.
No, I don't know about that, but they're definitely not resistant to something called genetic drift, where yeah, if some stochastic event happens, the small population can be easily wiped away at one go. But yeah, so that's like the genetic drift. So the population increase might be very variable in a small population. In a larger population, the population increases definitely. You can read upon this genetic drift and stuff like that. So I think you've been taught that as well. But anyways, what's the answer for 13C? C, yeah. So this was the paper done by Robin, Whitebelly Sholakili and Nilgiri Sholakili. He happened in the 2015 paper. He happened to bring in some taxonomic changes. So the Sholakili was called shortwing for a really long time. Supposedly of the same family as the shortwings in the northeast, but after this paper, it was found that these two birds were from a totally different were totally different from the northeast populations, and also that the birds that were apart from one particular barrier, they were like very different from each other, and the time of divergence was basically 5 million years. Yeah.
Sir, with regards to the mountain goat example you provided, was that the Pyrenean ibex by any chance?
Yes.
It was found in the Alps, right, sir?
I suppose. Yeah, one in a very temperate climate, the other was slightly tropical or subtropical. The population, basically. How was the period determined? Interesting question, Dr. Vedang. How was the period determined? So basically, by statistics.
Sir, was this by the molecular clock?
You're talking about molecular clock, divergence by molecular clock. Yes, Dave, you have something.
I think there was two tectonic plates crashed and created this barrier, and that's how these two populations got separated 5 million ago. I don't know. I thought I read it somewhere.
Well, actually, this is the Western Ghats, which is
Right.
a lot
Within
older
Western
than
part, the Nilgiris and the other part. So there is a barrier which was raised because of some clash. I think I read it in that lecture only.
No, geological basis wasn't used as a to determine. Molecular clock is used to determine the So divergence probably, molecular clock might be used, but basically what is used is how different the alleles are. So statistics is used like you have some 100 alleles, I have some 100 alleles, and if 95% of my alleles are different, we both share only 5 alleles, then we would be this. Our relationship would be this old. But if we have some 80 alleles share, that means that our relationship would be much recent. Our ancestors would be very recent. Do you get the analogy that I'm trying to tell you over here?
Yes, sir, but I think you just described the molecular clock.
Yeah. But molecular clock is using the mutations, right? The number of mutations. There is like a particular rate of mutations that happen, and
yes, sir. It's. I think if I remember the definition correctly, it's that mutations tend to happen in a certain pace, and depending upon the on the pace, it's possible to trace the history of when things diverged or species might have come up. It's something
but that
like
is mutation, right? I'm talking about the number of alleles shared. No, there's a difference, right? I don't know.
Yes, sir. I guess it focuses on mutations, but I guess the method used is somewhat similar. The timing.
I don't know. So that's why I don't want to go much further into it because I have never done it. Probably you can read up on the paper. But yeah, what is the answer to this one? It's 2015 paper by Robin and I think Pooja Yadav. I can share that with you. So basically, if you have done even a little bit of statistics, this particular term is very similar to ANOVA, where one is looking at the variance within the population and variance between the population and seeing how different they are. If the variance between the population is much higher than the variance within the populations, then that would mean that there is a significant difference between the populations. So that is something very similar to what this uh, this particular term means. The answer is A, FST, or it's also called F statistics, which is again very similar to ANOVA. If you want to read up on ANOVA and F, yeah.
Sir, I think this is what I mentioned. I read somewhere either about the kakapo or some other species which is endangered in an island, but does not suffer from immense amount of inbreeding issues because they are resistant since they are naturally low population.
So they do have, they are highly inbred, but they don't have deleterious alleles. So they are okay, is what people say. Yeah.
Is there what kind of adaptation allows them to withstand the effects of inbreeding, sir?
It's not an adaptation. It's just that actually I'm not sure. Might have to read that paper again. But what do you think?
Well, I think since they are naturally low population, the number of bad traits are somewhat on the low side, which allows them to, which is why they can suffer. They can be so inbred and not have any issues.
So you're trying to say that because they're anyways at a low number, uh, yeah, I'm not sure how, but yeah.
Since they're anyways a low number, the number of harmful mutations might be lower. At least I'm just guessing, sir. I have no real background
Yeah,
in
I
this
know.
topic either.
I'm not. Yeah, I'm not convinced with your argument, but yeah, I'm not sure. But the deleterious, I mean, mutations are sorry, deleterious. Why is it difficult to pronounce this word? Deleterious alleles are low, so that would mean that yeah, the population would survive. But how is it low? Yeah, that is something that the paper must have speculated. Anyways. How is deleterious different from recessive? Um, Recessive is just the opposite of dominant, right? It need not be deleterious, as far as I understand. No? Yeah. Maybe since they have inbred for long enough, they have already expressed recessive deleterious genes in the past. Long enough. Okay. Yeah. But, I don't know. I'm still not convinced. Probably you guys actually should read that. I mean, the Kakapo paper mentioned in Robin's lecture, and yeah, probably that would give you a better answer. Some versions of a gene can be lost due to random chance, and this is more likely to occur when populations are small. Additionally, smaller population means that individuals are more likely to breed with close relatives.
I just Google the answer, sir.
Answer? This
Well,
is not.
that's not the answer. Google the question, so that's what I.
No,
That
but
still doesn't
yeah,
explain
it
it.
doesn't explain anything. This just tells you what genetic drift is, and again, inbreeding. The first part of your statement is genetic drift, where genes can be lost due to the random chance. And the second part is basically telling about inbreeding. Genes which don't express will be called recessive. Huh.
I can paste the link where I found this, sir.
Question 16 is confusing. What is not a method? Oh, I'm so sorry. What is a method? What is the method to quantify genetic variation?
Yeah. No.
Yeah.
Because if I have an option as D, then
No, none of the above. It should have been none of the above. Yeah. But
okay.
anyway, sorry. Yeah. What is the method to quantify genetic variation? All of them actually are the methods to quantify genetic variation. No wonder
Okay.
you were
So,
not answering the question. I was like, nobody's answered any. Huh.
okay. So the question is, what is a method?
Yeah.
Okay. Not is not there. Okay.
Captive breeding effect of something. Okay. I'll have to click on it. Is there a link that you sent? It doesn't say anything.
Yeah, a second, sir. Doesn't say anything.
No, it just takes me to the Purdue University website.
Sorry, sir. Just a second. I posted it now, sir.
Yeah, that's what I'm saying. I mean, what you posted is the effect of small population size, and what it's showing is basically the harmful effects of small population size, but still doesn't answer what we are looking for. Why were the deleterious alleles not found in the small population of kakapo? Sorry. Do you get my point? Do you get the difference, Govind?
I get, sir. I mean, I'm having a hard time actually finding a suitable.
I think we can just Google that particular paper. I am not sure where it was. Okay, just a sec. Kakapo. Where was that paper? Yeah. So the Kakapo paper, it dissects et al. To 2021. Do you just a sec? Let me get dissects et. So the paper is, I'm posting it over here. I hope you admixture between so they talk about something called purging to take place, similarly remain consistently small enough for purging to take place, potentially challenges the benefits of okay, what is purging? Genetic purging is the reduction of the frequency of a deleterious allele caused by an increased efficiency of natural selection prompted by inbreeding. Purging occurs because many deleterious alleles only express all their harmful effects when homozygous present in two copies. During inbreeding, as related individual mates, they produce okay, blah blah blah. Put another, natural selection removes inbred children and their offspring with deleterious alleles from the gene pool, purging the deleterious alleles. Purging reduces both the overall number of recessive deleterious alleles and the decline of mean fitness caused by inbreeding. The term purge is sometimes used for selection. Okay, basically what's happening is all the offsprings that have deleterious alleles are dying. Purging was a hypothesis, isn't it? Yeah, purging was a hypothesis, and that's what they hypothesized. But yeah, look at the paper further, I suppose. Yep, the answer for this is all of the above. What is the method to quantify genetic variation? All of the above can be used for quantifying genetic variation, pairwise difference between the sequences. This is the average pairwise difference, and then segregating sites, like how many SNPs are changing in different sequences. Then heterozygosity can also be measured.
Sir, I am not able to open the link. Is anyone else able to open it?
I also can't open it.
No, even I could. Maybe if you could just send us the name of the site or something.
Yeah.
I think that URL, when it's copied, it doesn't retain that.
There are also
Yes, paper topic may be useful with authors.
um, this is the exact topic. This is the exact topic of that paper: genomic signatures of inbreeding in a critically endangered parrot, the kakapo, 2021. Probably you can Google Scholar it and get the paper. I mean, for me, I think my laptop is connected to the institute internet, so I was easily able to access the paper. If you want, I can download this paper. I can easily download that paper. Do you want the downloaded copy? Or could you access it? Okay, I can't send you the downloaded copy, at least over here. Are you guys going through the paper, or did you find the paper?
I could open it.
Yeah. At the end of that paper, there is this mention of purging in the discussion. So I think page number 9. No. Page number 9. The left part, sorry, the right first paragraph. Somewhere in the middle is through a combination reason for mainland kakapo has higher mutational load than individuals from Stewart Island, suggesting that deleterious alleles may have been removed from the Stewart Island population through a combination of genetic drift and purging. Limited evidence for inbreeding depression in our study may potentially also be explained by the dynamics of purging and alleviation of some of the effects of inbreeding. Yeah, possibly. Might have to go through the paper very carefully. തീർന്നില്ല. All right. So Ishtiaq's paper says that Asian gyps vulture, even after a population decline of 97%, still showed like a low level of inbreeding. But there was a major caveat that Robin has mentioned in the lecture, and that's what the answer is basically. Most of the samples, or actually all of the samples, were collected from captive population. So what's the problem with collecting samples from? How is that a caveat? Any idea?
There's a lot of human interference to prevent inbreeding.
No. That's not what Robin. I mean, that's not what the hypothesis was. Captive population. Any other idea? Like why was it a problem? These are long living birds, huh? Very, I mean, they live a long life. So, what could have been a problem? So basically, it could happen that the captive population, the birds that were captured were way before the time when diclofenac, that whole incident happened. That was in, I think, 1980s or something. So I'm not quite clear about the timeline now, but whenever that whole event happened, probably the population might have been captured before that. So that could mean that the population was way more diverse before that, but after the population crash, uh, yeah, I, the paper wasn't sure that if the samples collected were from after the population crash, which would actually tell you how inbred the population is. But yeah. Dr. Vedang, are you asking about the finding of the last study or this study? According to Brower and Falling, can be a general explanation for interspecific variation in extra-pair paternity in socially monogamous species. Okay. Oh yes, the one on vultures. The one on vultures, the finding was there is, I mean, even though the population crashed like 97%, the finding says that there isn't much inbreeding seen, other like low levels of inbreeding in the in the genetic samples that they have collected. That was the finding. I suppose I can also give you the link to that. Yeah. Ishtiaq 2014. Farah Ishtiaq. Huh. This is the name of the paper, Dr. Vedang, if you're asking. And according to Brower and Griffith 2019, following can be a general explanation for interspecific variation in extra pair paternity in socially monogamous species. So basically, interspecific variation would mean that there are different differences of EPP that is found in different species, and people have come up with a lot of theories why EPP is found in certain species and why not in certain species, or why it's more in certain families and why not in certain families, why less in certain families. And there have been different kinds of explanation given, like yeah, some of the papers have given latitude as one of the explanation, like latitude, like birds in tropical areas have more EPP, and birds in temperate areas have less EPP, or the opposite. And life history or ecology. Ecology would mean that birds that live in the reed, that nest in reeds have more EPP, or that nest on the trees have more EPP. So that is the kind of explanation a lot of people have come up, like there are a lot of papers. And yeah, the last two questions are just to point out that there could be studies that say very different things. But anyways, question 18, Brower and Griffith says this 2019, and answers, mostly people have said C or B, but the answer is actually D. So Brower and Griffith 2000 paper, if you check, I can just if you check that paper, you'll realize that what he's trying to say is none of the none of the variables actually explain any kind of none of the sorry none of the variables explain any kind of this interspecific variation in EPP, and it's by chance, or at least it's not explained, or it's not explained by any particular variable. So let me just find. You have the name of the paper in the this thing. You can find it from there. Extra pair paternity in birds. It's a 9. Yeah. First degree relation, I guess. What do you mean by that, Dr. Vedant? So if you read this paper, the paper says that there is no relationship, or at least there is no trend that can be seen for the presence and absence, or for the degree of variation of EPP across several species. Yes, Dave and Govind. I suppose Govind, you can go first. You've raised the hand first.
Yes, sir. Well, if I'm not mistaken, genetic monogamy is very, very rare in birds and animals in general, right, sir? So extra pair paternity in socially monogamous species is more like the norm rather than the exception.
It's not very rare. If you read the paper, you'll realize that there are quite a few species where actually there is no extra pair paternity, or there is no evidence of EPP. I think 30%, like 70% of the birds show EPP, but 30% of birds still don't show EPP. So it's not a norm as such. Did you get my point, Govind?
I guess, sir.
Read the paper. And Dave, you had a question?
Yeah, in this question, why is it called interspecific? Should it not be intraspecific?
Intraspecific is within the species, within a particular, within one species. Interspecific is between species.
So no, like extra pair paternity is within the same species, right? It is not happening across species.
No, it is within the same species, but okay. So, uh, but the variation that you see across all the species, like there are some species which have very high EPP and some species which show no EPP. So why is this variation? They were trying to find out if this variation can be explained by some variable, like some biotic variable, like
Okay,
climate,
okay.
latitude.
I misread the question. I thought
Yeah,
it
yeah.
was like some quality, which is it's a specific variation.
Yeah,
Okay,
yeah.
thanks.
All right. So, last question. Okay, so this Valco et al. Is not 2019, it's a 2021 paper. The paper is cited in the bottom of the slide. It's a 2021 paper, okay? So it's more recent than the 2019 paper. So actually, it would be nice to look at both these papers. They have very Valco et al. 2021, the frequency EPP decreases with latitude, So the latitude increases EPP. So basically, they're saying that tropical species have more EPP, and temperate species have less EPP, and increases with distance from breeding range boundary within the species' breeding range. Distance, okay. So that would mean the migratory birds have a more chance of EPP. Is negatively associated with generation length. So the longer living species have less chances of having frequencies of EPP, and the pair bond duration among species. So um, yeah, so more time the pair is bonded during the breeding season, as they have less chances of EPP according to them, or decreases with latitude as assemblage level. So assemblage level is basically there's a Palearctic assemblage level, like biogeographical level. There's a Nearctic, like different kinds of like global biogeographical levels. Okay, let's see what the answer is. So the answer is all of the above. There is not one particular reason. There are, like, the paper says that all of these reasons are responsible for the frequency of EPP, or basically how Dev says interspecific variation in extra pair paternity. So yes, Govind.
What does option C mean, sir?
It reduces with latitude, but even within like biogeographic regions, like Palearctic would mean that or Oriental would mean Indian subcontinent and some parts here and there. Palearctic would mean that everything above the Indian subcontinent, like the then Eurasian. Then Nearctic would mean that the So these are these assemblages of assemblage level. These are the like assemblages of different communities of birds. So even within them, as you go towards the latitude, the EPP decreases. So it's that trend is shown at all these levels rather than a global level. I mean, it is shown at a global level as well, but even at a smaller level, it shows the same trend. I hope you got it.
Oh, so even the decrease in latitude is present even within one particular biogeographic region instead of simply the large scale, both
Yeah.
small scale and large scales. What you're
Yes,
saying.
yes, yes, yes. All right, Dave Ball is saying good night. That's the last question. So we can discontinue unless you have some questions, some more doubts to clarify.
Just one, sir. What is the EPP level of a hornbill?
Wow. I think because they let's just look at this decreases with latitudes and increases with distance. So basically, hornbills are more tropical species. So definitely, according to the paper, it should have more EPP. Then negatively associated with generation length and pair bond duration, which is both very high for hornbills. They are long-lived species and they also pair bond for a very long time. Then decreases with that doesn't explain much. So both A and B have opposite this thing, but I think check the last paper that I was talking about. This one, Brower and Griffith, they have showed these trees, like what phylogeny trees, explaining where extra pair. Wait, I'll just show you where it is. Brower and Griffith. Is it Brower and Griffith? No. Extra pair paternity. Yeah. So parrots, falcons, hornbills, they have some. Okay. Okay, I can't send an image, but if you look at the paper, you'll find that there is in the hornbills it shows very less. Wait, where did it go? Yeah. So hawks and eagles show a lot of EPP, but hornbills and woodpeckers, it's not like it's zero, but there is some EPP seen in that particular family of birds.
So it's very low for hornbills.
Are the links to these papers handy with you? I'll do one thing. I can't send you the PDF. Okay. The link is here. Okay.
Sir, could you post a PDF in the discussion forum then?
No, I can't. Discussion forum. Oh, no, I don't want to do that. That's a very public forum. No. I mean, the whole copyrights and stuff will come up. Brower, that link is here. And we have, and the other one, I can just check if I have it. Okay, I did download it. Where did it go? Anyways, I can't find it now, but just Google up that name of the paper. No, what is it? What was the paper called? The macroecology. Yeah. Okay, got it. Here, the second paper. Are we having such discussions again? No, this is the last discussion, Dr. Vedant. I think this was the end of it, and I mean it. Yeah. So, anything else? Yeah, it was nice talking to you, Vedant. Like, good drama you are sometimes. Thanks. Anything else? Anyone else? Before I close the session.
Thank you for all the sessions.
Yeah, you're welcome, Bruce Bay. And I think my email ID or whatever is there on the discussion forum in case you want to reach out. But otherwise, you have, yeah, you have all those senior ornithologists on the group. They, I mean, you can reach out to them directly if you have any questions regarding their particular subjects. But yeah, it was all, yeah, it was really wonderful interacting with all of you, like Vedant says. And thanks. Thank you, Harini.
Thank you. Thank you.
Yeah, thank you, Astagmat. All right, see you sometimes else.
I'll really miss the session, sir.
Thank you, Govind. All right.
Thank
Yeah, thank you, Vikas. Bye. Good night.
Good night, sir. Thank you.
Good night, sir.
Bye.